Hey everyone, my name's Amanda, and as many of you know, I work in the cutting edge field of genomic medicine, and I spend my days looking through DNA tests, interpreting them from a clinical perspective for clients, but also working worldwide with clinicians to teach about this cutting edge field and helping them understand how to interpret these tests and change the lives of our clients. What I want to share with you here in this brief video today and over a series of little snapshots are some of the insights that I have from working day in and day out and around the world with DNA tests. So let's get started and just keep checking in, and I'll be dropping in these observations as often as I can. So the first thing I want to share with you is something that every single person I work with, whether you're a client of mine or I'm working side by side with you as a clinician, is this. Genes are signposts in a nutrition or medical toolbox that tell us where to look and what questions to ask. They take the guesswork very often out of what's wrong because they help us think about what's really under the hood of our operating system. What's under the hood of a biochemistry that's not working efficiently. So genes are signposts. They're not diagnostic, they're not predictive, they're an important part of a toolbox we use to help piece together the story of your health. Here's observation number 2 from my work in genetics. One gene does not derail your health, your operating system, or your biochemistry. Here we're talking about genomics. We're talking about variants that we call SNPs, or single nucleotide polymorphisms. These gene variants determine something about who we are. They determine our traits, the color of our eyes, whether we have frizzy hair or straight hair, things like that. One SNP does not derail your health. What we know in genomic testing and what I and my colleagues do on a day-to-day basis is we look for patterns in these gene variants, so these SNPs, to tell the story of who you are and to tell the story of where some of your biochemistry, which is your human operating system, is working efficiently or inefficiently. So let me give you an example that is very commonplace that we see coming from members of the public and concerned families. And they'll say, I've got MTHFR. And our answer clinically to you or anyone having MTHFR is, we all have MTHFR. Genetically, we look at MTHFR for two different perspectives because we look, at least from the literature and the science right now, at two different forms of the genes, this gene, and how the variants within this gene, when they're added together, can be part of a story about how you handle folate in your body. So each one of us has MTHFR. Each one of us has two forms of this MTHFR, if you will. But MTHFR is not the only gene that we consider when we're looking at how your body handles folate, which is a function of this gene. We look upstream to other forms of genes in the folate cycle, such as MTHFD, MTHFS, and also, if you will, downstream to how this gene interacts with another gene, MTR, and another gene, MTRR. So know that one gene never works by itself. It's part of a team. Here's tip number 3, or insight number 3 from my work in genomics and what I see on a day-to-day basis when I look at DNA testing. I see a lot of individuals who have a gene that's deleted in their detoxification or waste management system. This gene is called glutathione S transferase, or GST, and it comes in different forms like GSTP1, GSTM1. Frequently, I will see a deletion in a member of this family, which means it simply isn't part of the detoxification process in your waste management system. Some of you know that we think of detoxification in 3 stages: stage 1, stage 2, and stage 3. Glutathione S transferase is active in stage 2. It's rarely a gene that we would look at and say, that's it, you can't remove toxic byproducts from your body. But when it's missing, it's kind of a little bit of a hamperer on the system, of the detoxification system. Means that your body has to work harder. You rarely have one gene variant in detoxification. Ultimately, you'll have several, and it's the totality of all of those genes, whether it's in the first stage of detoxification or the second stage, for example, that we're looking at. What I will tell you in clinical practice is that people who have the GST deletion, usually the GSTM1 deletion, along with other gene variants in stage 2 detoxifications, actually have challenges, real challenges, in the efficiency of removing waste products from the body. Waste products are byproducts of metabolism, pharmaceuticals, hormones, toxins, things that we're exposed to every day. So detoxification is very real. And if you ever hear anyone saying your body has a natural ability to detox, you don't need to support detoxification, I would say to you that the body does have these systems, but genetics will determine how efficient they are. And therefore, some people need more help than others, period. We know this through genetic testing. Genetic testing tells us where to look. We validate the efficiency of genes through functional testing. So we can literally see how well people detox or not. That is the great power of genomic testing. What else do I see when I'm looking at genetics on a day-to-day basis and I'm working side by side with people like yourself and clinicians around the world? One of the things that genetics has me looking at every day is not just DNA, but DNA takes us to validation in lab testing. So what I'm getting at here is a lot of times people may genetically have a predisposition to a process called poor methylation. So methylation, I call it the heartbeat of human biochemistry, because this important operating system in our body produces something called methyl groups. Methyl group is one hydrogen surrounded by 3 carbons. Actually, it's the other way around. It's 1 carbon surrounded by 3 hydrogens, and I'm just going to let you hear me make that mistake. I talk about it every single day. So a methyl group, carbon, 3 hydrogens, right? Otherwise, we wouldn't be human. So methyl group, we like to think of being kind of an activating hook that we sling around the body, if you will. It activates hundreds of enzymes, hundreds of processes in the body. Super, super important. So what is it that I observe here? When I am looking genetically and I suspect that somebody has a poor methylation ability, I don't just say, you're a poor methylator. What I do is I order lab testing or request lab testing so I can look at not only the efficiency of the methylation cycle, but whether you or anybody I'm working with has sufficient number of supporting nutrients. Supporting nutrients for methylation include pretty much all your B vitamins, very specifically B3, which is niacin, B2, which is riboflavin, B9, folate, B6, cobalamin, B12. So